Hello, I'm Sadia. I'm a PhD student of Illinois State University. So in my poster presentation, I will introduce you with the uropathogenic E. coli specific transcriptional regulator. When a pathogenic bacteria enters the body, our innate immune cell, like neutrophil, engulf the pathogen and kill it by generating reactive oxidants. Neutrophil has antimicrobial enzymes, NADPH oxidase, that reduce molecular oxygen and generate reactive oxygen species. Neutrophil also has myeloperoxidase, or MPO, that catalyze the formation of reactive chlorine species. These reactive oxygen and chlorine species are highly toxic for the cell. However, the most potent and abundant one is the hypochlorous acid. Just a side note that hypochlorous acid is also an active ingredient of household bleach. So therefore, in my talk from now on, I will use the word bleach instead of hypochlorous acid. So as bacteria encounter bleach stress upon entering the body, so I was wondering how bacteria respond to this stress. So for my study, I used several species of E. coli, and I have found that uropathogenic E. coli, or UPEC, are more resistant to bleach than commensal or non-pathogenic E. coli. For further verification that this resistance is UPEC specific, I used another strain of E. coli that is intestinal disease causing, known as enteropathogenic E. coli, and I have found that this enteropathogenic E. coli's response to bleach is similar as commensal E. coli's sensitivity. So this confirms that UPEC is more resistant to bleach than other E. coli. Now the question comes up that how UPEC defend this bleach stress. So we performed the transcriptomic analysis and we observed that several UPEC-specific genes are upregulated under bleach stress. Among them, 3 genes looks very interesting to us as they are located adjacent to each other, and one of them encode a putative transcriptional regulator we named as RCLR2. And I have found that this regulator controls the expression of 2 uncharacterized genes, RCLD and RCLE. So when we deleted the RCLR2, we observed that now the mutant UPEC strain is more resistant to bleach than wild-type UPEC. However, the interesting one is when I deleted the RCLE, one of the target gene of the regulator, and I have found that RCLE deletion UPEC strain is highly sensitive to bleach, a sensitivity that is correlated with the commensal or non-pathogenic E. coli's bleach response. So as RCLR2 control expression of such a crucial gene, I was focused to characterize this RCLR2, and I have found after performing biochemical and molecular analysis that RCLR2 functions as a transcriptional repressor, which binds to the promoter DNA under non-stress condition and derepress the target gene's expression. However, upon bleach stress, it dissociates from the promoter and derepress the target gene's expression. And this is evident by our EMSA experiment, where we observed that in absence of bleach, there is a protein-DNA interaction. However, in presence of bleach, this interaction disappears. And it again reappears when we restore the reducing environment. So what happens with RCLR2 upon bleach stress is that they forms intermolecular disulfide bond. And this is evident by our SDSH analysis, where we observe that high molar mass band formation upon increasing bleach stress, and this band disappears when we restore the reducing environment. So cysteine amino acids are particularly the major target of bleach oxidation, and I have found that 2 cysteine are responsible for RCLR2's redox regulation. Because when we mutated these 2 positions of the cysteine, it was unable to complement RCL2's function. And this is evident by their increasing resistance to bleach compared to the wild-type complemented RCLR2. And with this, what we have is that UPEC is more resistant to bleach than other E. coli, and RCL-R2 is a bleach-sensing transcriptional repressor of UPEC that has cysteine amino acid as a redox switch. And RCL-E, a target gene of the repressor, is crucial for UPEC's bleach resistance. With this, I want to thank our lab group and my colleagues, and also our collaborators and funding sources. And if you have any question or comment, please feel free to email me or contact me on social platform.